afternoon, everyone. Thank you for joining us again this week for another episode of NHS Talks. It's very mild here on the East Coast, so I hope everyone's having some great weather out there. It's our pleasure to bring back Dr. Robert Green. A few years ago, Dr. Green was kind enough to present to our Boston chapter with regards to the genomic medicine and where the field is going and the research that he's been working on. And we thought it'd be great with everything that's happening in COVID. Certainly, we've heard updates from experts, epidemiologists, and virologists, and people in the medical field with regards to how things have been progressing, and with looking at potential vaccines on the horizon. But today we're going to take a different approach and educate you on COVID and the world of genomics medicine and how people like Dr. Green have been instrumental in researching COVID through the eyes of genomics and looking at what some of the kind of early stage discoveries are. Dr. Green is in the forefront here in Boston, but also not only here at Brigham and Women's and Harvard Medical, but in his own lab and his own research project, and is a very, very renowned figure in the field of genomics medicine. And we'd like to welcome him today. Dr. Green, welcome back. And assisting him will be Bethany Zettler, who works closely with Dr. Green. So Dr. Green, I'll let you take it away.
Thanks very much, Drake. And hello, everybody. I'm delighted to be speaking to you. I had the pleasure of speaking in person to a group in Boston not too long ago, I guess a year or two ago. And that was a lot of fun. Stayed in touch with some of you, and I'm delighted to speak to you again. We're going to talk today a little bit about how our program has pivoted to try to support COVID research. The focus of our talk is on the genetics of COVID vulnerability. And I'm going to speak today in a tag team with Bethany Zettler, who is a genetic counselor, also has a background in microbiology, and she's going to help me talk to you about one of the components of this, the vaccine component, when we get to that. But what I should say first is that never in my entire career have I seen the scientific world as one pivot to try to work on something with such energy, such cooperativity, such incredible persistence as I have seen from every single one of my colleagues in research throughout the Brigham, throughout Harvard Medical School community, throughout the world. And so each of us has tried to do our small part. What I'm going to tell you about today is obviously a very small part of the puzzle, because COVID reaches across so much: epidemiology, virology, immunology, vaccine production, cardiology, pulmonology. So many people have brought their talents to bear. So I'll be talking about a narrow sliver of the topic, which is the genetics of COVID vulnerability, because that's what I've been able to try to contribute from my position as a geneticist. Just as a reminder, I direct something we call Genomes to People, which is a program dedicated to understanding how we can use genomics to do a better job in medicine. Why is it that your doctor hasn't already said to you, haven't you gotten yourself genome tested, so that we can know if you're at risk for cancer, we can know if you're at risk for heart disease? And we all think this is coming, but it's had a really hard time getting here. And my research is dedicated to getting it over that last mile so that we implement it into the practice of medicine. Now, most of you know well the timeline of the COVID epidemic, but here it is, just summarized, all the way from December 2019, doesn't that seem like ages ago now, to basically the last few days, when there have been almost 50 million cases confirmed worldwide and 1.2 million deaths. And one of the things that's really interesting about COVID is how it spreads all over the world, how it spread from China to Italy, to the rest of Europe, to the US, down to South America and so forth. We're going to get back to that in a minute. But let's talk a little bit first about how COVID works. COVID is an insidious little genetic hijacker. it actually is an extremely efficient little pathogen. It's got this capsule, and it's basically got RNA stuck inside this capsule. And the RNA is very, very efficient. It's a tiny little piece of genetic material. And what it does is it comes into your body, usually into your nose or your throat, or sometimes perhaps your eyes. It connects onto this receptor that's sitting there being supposed to be used for something else, but it's figured out how to hijack this receptor. Then by connecting onto that receptor, it goes into your cells where it replicates itself, makes thousands of different copies, and then spreads to the rest of your body and of course, when you breathe, out into the rest of the world. Now, the virus itself has, even with that simple piece of DNA, it has a surprising number of variations. Little changes that, the COVID that's in South America is not necessarily the COVID that's in Italy. And this has made it really interesting to track the spread of where this goes. So, there were some colleagues of mine in Finland, and they could tell that their particular strain of COVID came from someone who was in Italy because of the genetics of the virus itself. So it's kind of like a fingerprint or a footprint that you can trace as it spreads all over the world. And I thought, since this is the Hellenic Society, you might like to see the ways in which these genetic footprints have transmitted across the country of Greece. And indeed, they have come from everywhere. So there are some that have come from Italy, some that have come from Serbia, some that have come from Bulgaria, and many other places. So there isn't just one stream that has come to Greece. And at this point in the world, with the world so interdigitated with itself, it's probably true for many, many areas at this point in time. There is, of course, new cases per day in Greece is going up as well as it is in the rest of the world. Now, that's just a very brief overview of the genetics of the virus. But now let's turn to the genetics of human beings who catch the virus. And, as a geneticist, when this broke out, I looked around and said, well, how can I contribute? And the main question that I thought I could be part of the team contributing was, why is it that some people have very severe symptoms or even death, and other people have very mild or no symptoms? Is this due to something in their genes? Okay, that's a tractable problem I thought maybe we could collaborate on. Now, as with so much in our life these days, there's a fair amount of misinformation. And what was immediately evident as the pandemic broke out was that some ethnic groups seemed to have a worse time of it than others. In the United States, African Americans and Hispanics to some degree did worse than their white compatriots. If you look here, you could see that Black people were dying at 2.4 times the rate of white people. So you would be forgiven if you thought, oh, well, maybe there's something about the genetics of Black people that makes them more vulnerable. And that, isn't necessarily an outrageous hypothesis, but I can tell you right now, we've looked into that. Many people have looked into that. And when you control for socioeconomic status, access to healthcare, and all the other sort of socioeconomic things that are correlated with being Black in this country, it simply isn't true. In fact, as this Scientific American article bluntly said, it's racial disparities, not genetics, that are explaining why more Black Americans are dying of COVID. So I get this question a lot. I've talked to Asian audiences. They're very keen on whether, Asians are somehow more vulnerable, more protected. It doesn't seem to be at that level. But early on in the pandemic, our teams here in Boston, particularly our teams at the Broad Institute, collaborated with teams in Finland to create a worldwide COVID-19 host genetics initiative. Again, the host genetics means us as the host for the virus? What is it about our genomes that might make us more vulnerable? For instance, we already knew that ACE2 receptor that I told you about, that's where the virus latches on. We already knew from other research that there were lots of genetic variations in that. There's at least 17 different genetic variations in just that receptor protein. So it would stand to reason that some of them would bind to the virus more readily than others. And indeed, that was one of the earliest observations, that some people with a particular variant in that protein bind tighter or less tight to the virus. That's the kind of thing that you could ask, well, how do you use that? You use that because it's not because you're going to try to identify vulnerable people. You use that because it gives you a clue to creating a virus, a vaccine, or a treatment that could take advantage of that knowledge. Now, this initiative has now got more than 1,000 scientists from all over the world. You can see some of the people who were registered early in this, but there are now teams from all over the world. It's been an actual joy. Nobody's worried about funding. Nobody's worried about credit. Nobody's worried about anything. The first paper came out here, and you can see nobody insisted their name be on it. It was just from the COVID-19 Host Genetics Initiative. And so this has been one of the purest, most collaborative experiences I've ever had. And essentially the question is this: if we take people, let's say 100 people who get COVID, we know that some of them are going to actually die or have very severe disease. Let's say these people in red. Some of them, like this yellow person here, is going to have an unexplained resistance to the infection. Some of them are going to be young and they shouldn't be so severe, but they're going to be very severe. How do we sort that out? And I thought I'd give you a little inside view of a research project like this, because it's more complicated than you might think. First of all, we gathered a team together just in our institution of myself, a couple of people who are really into the genetics of common diseases, Jordan into the genetics of schizophrenia, Beth into the genetics of rheumatoid arthritis, a couple of our genetic counselors who manage projects, the bioinformatics guy for all of Partners and now Mass General Brigham Healthcare, one of the best diabetes geneticists in the country, one of the best cardiologists in the country, the guy who runs our biobank, the woman who is an infectious disease expert and is defining these cases, and the guy from Broad Institute who is an expert on sequencing. And that was just the beginning. There are like a dozen additional people who have jumped into just our version of this project, just trying to figure out how to do this. Next, we had to define what was it we were comparing. And we decided that the most important comparison we were interested in was those people who had severe disease, i.e. they needed, as defined by needing respiratory support, oxygen, intubation, or who actually died. So that was one group, versus people we knew caught the virus but did not require respiratory support, basically everybody else. So we said, okay, these are going to be the two groups that we're most interested in. Then we started collecting these out of our electronic health records and out of our biobanks, and where we had people who had donated DNA. Now, remember, you can't really build this up quickly. You know, you're in the midst of a crisis in a hospital. It's very hard. Somebody's on a respirator in this group. What do you go up to them and say, hey, can I take a DNA sample because we're kind of interested in this research project about genetics of COVID? You know, the poor person is struggling for their life. Luckily, we had over 100,000 people who over the past 5 years had donated for any reason their DNA to the Mass General Brigham Biobank, including some of you guys. And those people's DNA was broadly consented, so we were allowed to use it for anything we could tie into their medical record. So next we went into the medical record and tried to find the people who had positive tests for COVID, but who were intubated or needed respiratory support versus those others. And this is basically what every group did around the world. And it was harder than you'd think. At first, the first couple of weeks, it was like, 100 people here and 150 people here. And frankly, to run these studies, you need thousands of people. So thank goodness we were putting these together with people who were doing this all over the world. And I can tell you that we've already, when you see one of these lines pop up, that's a very exciting result. And I can tell you we've already got some very exciting results. These are the results as of just this week. And every week we're gaining more and more results. Let me show you some of those results. We found an area on chromosome 3 that involves 6 specific genes. And we found that people who are carrying one variations of these genes are 3 times more likely to get a severe illness than others. Now that's a clue. And when you look into what those genes are, you find that one of them has to do with the cell surface receptor on this very same receptor where the COVID guy latches on. You find that others have to do with the immune response. And indeed, it's part of the theory for why people get into such trouble with COVID and have respiratory failure and die is because their own immune system attacks them when it's trying to attack the COVID. We found other genes that seem to be associated with something called interferons, which again mediate the immune response. And this is why some people have been trying to counterintuitively knock down the immune response when people get into trouble, because it's your own body that's attacking you and making things worse. There's also an odd fact that I thought you would enjoy here, which is that this little cluster of genes on chromosome 3 is actually part of the genome that's come down to us from Neanderthals. Now remember, Neanderthals intermixed with Homo sapiens 60,000 years ago. And there's very little Neanderthal DNA in us. It's 1 to 3%. But strangely, this particular region seems to be part of the Neanderthal inheritance, which is kind of cool. So we've begun contributing as part of that giant consortium. We've also contributed because, for example, this is an article that I published back in June, which said, look, folks, if you had the knowledge of these 30 or so genes, we could take care of you better in the hospital. If this was already in your medical record, we could do a better job of taking care of you. So we've been contributing in our own modest way to the fight against COVID. I'm going to ask Bethany to talk to you a little bit about vaccines, because everybody's excited about vaccines right now, and she knows the vaccine data better than I do. So Bethany, if you tell me when to move the slide, I'll turn it over to you, and you can talk a little bit about that.
Absolutely, thank you. So as Robert mentioned, this is obviously, a very devastating pandemic and situation for the globe as a whole. But it's been a really, really historic time for scientific innovation and collaboration. And one example of that is this vaccine development landscape. This is by far the fastest timeline for any vaccine that has ever been tried in humans. We have a promising candidate in late stage trials in under a year from the first case. And it's really hard to overstate what an accomplishment that is. Next slide, please. So there are many, many candidates currently being tested in various stages of the trial pipeline. And these use all sorts of different technologies that I'll talk about in a minute. But the bottom line is that no single vaccine is going to be enough to fight this global pandemic. And so the landscape really is quite collaborative and supportive. It's very low competition. It's really, a pretty united scientific effort against this common enemy. Next slide, please. And overall, the goal of all vaccines is for our body to generate an immune response against a dangerous pathogen, a dangerous virus, without actually seeing that harmful infectious virus. And for COVID specifically, the part of the virus that our body needs to react to is the spike protein that covers the outside of the virus all around the surface there. And so our body will see that foreign protein, recognize it as an invader, and attack it. And so the traditional way to generate, an artificial response against this type of virus is to take the virus and grow a whole bunch of it and either kill it or severely weaken it, and then use this material as the vaccine. So this is how a lot of our traditional vaccines work. It's basically a dead version of the real virus. Your body sees it. It looks the same as the real virus, so we build an immune response against it, but it can't actually infect us. But this type of vaccine takes a really, really long time to grow enough of the virus to actually, use as a protective measure. So because we're in this, horrible pandemic, we're trying a lot of really interesting and novel mechanisms to actually generate that same immune response. And one of those especially cool mechanisms is using actual DNA or RNA, so using the genetic code of the virus to actually help our bodies develop an immune response. And this type of vaccine can be made a lot more quickly than the traditional vaccines. They may have fewer side effects, and they do seem to create a pretty long-lasting immune response. And this type of technology has been tried before for pandemics like Zika virus and Ebola virus, but this is the first time they've ever been used on a large scale. And so far, the data looks really, really promising. So it's an exciting time for that innovation. Next slide, please. And so this is just a little bit about the RNA vaccine mechanism, since that's the candidate that's looking best right now. So essentially, we look at the virus's genome, and that's just a genetic code. It uses exactly the same letters as our human genetic code, the codes for all of the proteins that make our bodies and keep us alive. So we can take out a little snippet of the viral genome that actually codes for that spike protein. So that's what we want our bodies to see and react to. And so we can basically take a step back from the actual protein and use the genetic code and put that little just snippet of RNA into basically an oily bubble and use that as the vaccine. So we inject that, that little snippet of genetic code into our bodies. Our body sees it, and we know exactly what to do with this little strand of RNA. We do it constantly, 24/7, in all of our cells. So we translate that into a protein, but it's not a human protein. It's because it's the code from the actual virus. So we're making the viral spike protein in our cells. And then this is excreted into the body, and the body sees that viral protein as, again, an invader, something different, something foreign, and our innate immune system actually responds to that protein without ever seeing anything that could possibly infect us or harm us.
Yeah, that's just so cool. Just to pause on that for a second, Bethany. I mean, you explained it very well, but we are actually harnessing our own cells to make proteins based on the genetic code of the invader, and then but we make a harmless piece of the invader so that our own bodies will recognize that and develop immune response against it. Is that right?
Exactly. Yep.
Yeah, that's so cool. Okay.
It's super cool. And then if our bodies actually do see the spike protein on the surface of the virus, if we ever happen to be exposed, we already have those antibodies. We remember seeing it. So we're able to react really quickly and actually kill it off before we ever become infected or get sick.
Awesome.
Great. Next slide, please. And so, yeah, this is the exact type of technology that is looking really, really promising in late stage trials, as I'm sure many people saw yesterday, this headline. So the vaccine so far looks like it's very effective. Obviously, there's still a lot of caveats. We don't know anything for sure yet, but this, again, the timeline is incredible. The new technology we're using is incredible. And so far the data is really incredible. So I think we do have reason to hope a little bit going into 2021.
Yeah, and this particular Pfizer trial is very interesting. They have been, first of all, extremely transparent. They've shared all aspects of the trial very openly. This one trial, they took a minimum or almost, I actually heard, no government money. So it wasn't being driven by any political agenda. They actually succeeded. It's been a real challenge to recruit a diversity of United States citizens into these trials. Pfizer went to the trouble to recruit, I think I heard 30 to 40% non-white participants, which is huge for building trust across the country that a particular treatment or intervention, will work in all of our citizens. So they're doing things really well. And so that's why I think you saw such a response to this headline yesterday, and such a leap in the stock market as well, because they covered many aspects of the viral science in ways that people found, were highly respecting. If we look at COVID deaths across the world, it's horrible. But if you look at the daily deaths of COVID, I just want to remind you that, compared to cardiovascular deaths and cancer deaths, they are still a relatively modest number of deaths. And these are the huge killers of our time. I say this because I want to pivot for the last few slides sort of back to our bread and butter research and tell you why we're so excited about preventive genomics. And the reason, in a nutshell, is that we think that by identifying people who are at increased risk for cardiovascular disease and cancer, we can help reduce these enormous number of deaths. Genomics used to be about rare disease. It is no longer. It is now about every disease. And that is the message we really have to get out there. We've led the way with a couple of studies that have become really impactful. The MedSeq project is the first trial to ever offer whole genome sequencing to ostensibly healthy adults, measuring the risks, the benefits, and the costs. And our BabySeq project pushed the envelope even further and has become the first study to ever offer genome sequencing to brand new born babies. This is a dream that so many people have talked about. They've said, at the beginning of our life, we should get a genetic blueprint of who we are, and then we'll have that information to help guide our journey throughout our life in terms of health. Nobody had actually, I know it's hard to believe, but nobody had actually looked for all this information and then given it back to adults or to families of babies before. So we did that. And what we found was really remarkable. Across all of these studies, we found that over 90% of us are carrying at least one, and sometimes up to 7 or 8, recessive mutations that can influence the health of the babies we have. Over 80% of us are carrying a marker, a genetic marker, that influences whether or not we respond well or poorly to a particular medication category. Over 15% of us, this is really unprecedented, over 15% of us are carrying a dominant mutation for cancer, heart disease, skin disease, hearing loss, eye diseases, you name it. But it's just way more common than we thought. And what's happening is that people are missing these. They're treating this hereditary heart disease as if it was regular heart disease. They're treating the hereditary cancers, the hereditary diabetes, as if it's regular. And that's okay in some circumstances, but in more and more circumstances, the treatments are different. And amazingly, over half of us are carrying what's called a polygenic risk, a summation of millions of genetic markers that puts us at high risk for garden variety heart disease, garden variety diabetes, garden variety atrial fibrillation, stroke, multiple sclerosis, and garden variety cancers. So all of a sudden, you can see that this really isn't anymore about rare disease, it's about all of us. And as we move forward with our health, as the cost of this genetic sequencing comes down dramatically, you can imagine that we're going to use this information at different points during the life cycle. We're going to look for pediatric diseases when we're just born. When we're about to have babies of our own, we're going to be interested in the recessive carrier conditions. We also are trying to do our part, just like we are with COVID, we're trying to do our part to battle against disparities in healthcare for our entire society. And this is particularly true of different racial groups within our society who've historically mistrusted medicine, mistrusted research, and have been disadvantaged at every point. And a lot of people have said to us, listen, you're developing genomics, but you're developing it for the wealthy and you're developing it for the privileged. And so we've gone out of our way to try to make sure that at least our research isn't perpetuating those disparities. I'm very proud to say, we have the first National Heart, Lung, Blood Institute project that will be returning genomic results to African Americans. We have the first National Human Genome Research Institute funding to subsidize the elective sequencing of minorities, people who are just plain interested in this, but may not be wealthy enough to pay it out of pocket. And we have the first NCATS funding, an entirely different institute at NIH, National Translational Medicine. We have a fundable score to start in December a new project to sequence Hispanic and African American newborns, baby-seq-2, if you will. So we're really proud of this. It's something we've invested a lot of time and effort. We've had some substantial support from the community to bridge this funding so that we can compete for these grants. It's been critical for us. We've also started the world's first preventive genomics clinic at Brigham and Women's Hospital. And we've co-opted some of our geneticists and some of our genetic counselors. We were doing it in person, but we can also do it now by telemedicine. And essentially, this is for people who want to be on the cutting edge of genomic medicine and who are basically healthy, but who might be worried about something. And they come in and at the moment, this is all out of pocket, but you can see here from our sort of menu that you can get a screening test that's made of 147 genes for $350. And that covers much of what you might be worried about. But if you really are interested in getting the whole shebang, you can get whole genome sequencing for somewhere between $2,000 and $3,000. And you can actually get up to 6,000 genes analyzed in these assays. And we can also, if you're particularly interested in having babies, we might direct you to a better value, which is getting a carrier screen for $250. If you're particularly worried about cancer or cardiovascular, same sort of thing. So we can really tailor this because there isn't a one size fits all. There are labs out there in the world, 23andMe, doing consumer genomics, color, Helix. They're all trying to convince people that, just take our one test. But medicine's not like that. People have different needs, especially for something out of pocket. They have different trade-offs for what they want to spend and pay. So that's pretty exciting. And that brings me to the end of the formal remarks. I deliberately left a lot of time because I'm really hoping that you guys will be interested in asking some questions and that we can get a bit of a conversation going. But here on this last slide, I wanted to make you aware, of who we are behind this program. We're just a group of people who are working together, who, just like you, have been struggling through pandemic. Now most of us are working from home, writing grants, writing papers, devoting ourselves to academic life. You can see a lot about our program here on our website. You can follow us on Twitter if you're a Twitter person. If you're an Instagram person, you can follow us there. And we'd be thrilled if you'd care to write us with any questions or personal interest that you have for yourself or your family. The community that Drake has introduced us to is very connected to the Brigham Harvard Medical School community, and we absolutely do not mind if you reach out to us with any kind of question. So let me pause there. I'll leave it on this slide for 1 minute, and I'll come back to it if you'd like me to later. But why don't you, if you'd like to, jot these down, and then I'll turn off the shared screen. And Drake, perhaps we can. You have some questions, or you can moderate other people who might have some questions.
Absolutely. First, yeah, thank you for leaving that up, and thank you, Robert and Bethany, for the presentation today. And quite frankly, appreciate giving our participants and members an opportunity to learn more and being able to directly reach out to you. And it's been interesting, seeing how the world has changed and really how science has changed in such a short period of time. But as I kind of start going through some of these questions, one of the first questions that came up is with everything happening around COVID this year, obviously being the greatest health threat. Have you seen an increase in people, individuals, families, whether they had someone that did get sick or they've been asymptomatic or never exposed, in terms of doing more of the some of the stuff you mentioned around donating samples and having their
I think
gene sequencing done?
I think it's influenced the public in a number of ways that don't really make it into the papers. But first of all, it's increased the conversation about science, and it's increased the conversation about genetics. I mean, everybody has heard, oh, this is an RNA virus. Well, what does that mean? And so, not everybody has dug into it, but even just hearing the terms has been remarkable. And I think it's increased the salience of genetics to the world. And of science in general. Hopefully people realize that science needs to be respected, despite what some of our political leaders have said, and that science is going to really make a difference. It's going to save millions and millions of lives in this pandemic. The second thing, Drake, is that people have really, really stepped up to contribute their own DNA. When we say to them, we're studying this, would you like to be part of the Mass General Brigham Biobank? They're like, yeah, absolutely, whatever I can do to help. So these are really relevant things for us. They make a big difference. They help morale. They help the bottom line when people donate. They help in so many ways. So it's huge.
And you referenced the biobank. I think you mentioned there's 100,000 people in it. Or volunteers. Whatever magnitude, though, that's probably a very small percent of obviously, never mind the population, but the population that actually goes through a doctor's office or has a health issue through a hospital. Yep. How does that 100,000 compare to other institutions across the country or the world with regards to, and overall, if you looked at it collectively?
Yeah.
Are we just scratching the surface?
Pretty well. Mass General Brigham has not been the very first healthcare system to create a giant research biobank. That honor in the United States went to Geisinger in central Pennsylvania. They've had like several 100,000 people. They did a deal with one of the big companies to sequence them in exchange for all the IP. And then a few other, Vanderbilt, a few other places Mayo Clinic have done some pretty impressive biobanks, but we are in the top 5 or 6 of, and so, we're growing this as fast as we can. We just got a big bolus of support from IBM of all places, who donated funding for some significant amount of sequencing of those people. Because it's one thing to get the people to donate their DNA, but then you need a couple $1,000s for each person at this point in time to sequence and interpret it. So it's still very expensive, but it's just so huge in terms of scientific payout.
And the people that decide to have any type of test or lab work done, what's the split between those that obviously someone in their family encounters an actual disease, a cancer or a cardiovascular event, and the genetic work is done as part of that to kind of trace family history, who else may have that variant, versus those that are just healthy consumers, but they want to be proactive. They want to see if there's anything kind of inside of them that they at least maybe should become aware of that could affect them or their family.
Yeah, great question. I think, so what happens is it's partly ascertainment bias. So we recruit from both inpatients into the biobank and outpatients. So if it's inpatients, they're people who are generally sick with something or having a procedure. And we go up to them, a research assistant goes up to them and says, hey, while you're here, would you contribute blood to the biobank? And they say yes or no. The outpatients, they can be coming in for a well visit, they can be coming in for dermatology, they can be coming in for anything. They'll be sitting in a waiting room and somebody comes up to them and says, would you contribute to the biobank? And they say yes. So we don't actually know till they get in the biobank. And then, somebody has to actually ask a question. So your question's a great one, but I don't think anybody has actually gone in and said, how many of our biobank patients have heart disease? It's a non-trivial exercise. We spent weeks with programmers, scientists, specialists, informaticians, simply figuring out who had COVID positive testing, who was likely to be positive and negative. We changed the definition after a certain point in time. We had to adjust for that. So we've done this for COVID, and I would tell you that out of those 100,000 people, first of all, we've only done genetic testing on about 40,000. So, and of those 40,000, I think we're up to 200 or 300 who by chance happened to get COVID. So they were in the biobank and then they got COVID. So that's the kind of scale I think we're operating at. And I would also say that related to the sort of general societal changes, Drake, one of the things that this has accelerated is telemedicine. You and I are sitting here on Zoom today, and this is how I see most of my patients now. Right. And we always knew this was more convenient and easier for both doctors and patients, but we have finally turned a corner and we're using telemedicine writ large.
As well as the samples you're collecting. I mean, they're kits that come to a person's home.
That's right. If anybody on this call wants to come to our Preventive Genomics clinic, they don't have to come into our office. They don't even have to be in Boston. We can see them by telemedicine, and we can send you a saliva kit to send the sample in.
Yeah. I do have one specific question regarding those of us that originate from the Mediterranean, specifically in Greece. Other than thalassemia, are there any other diseases in the Mediterranean or people of Greek heritage are prone to?
Bethany, you may have to help me out here. Beta thal is the one that I'm most familiar with. I don't know if there's any other kinds of sickle cells. Do you know of any others, Bethany?
So in terms of kind of the highest carrier frequency for severe childhood onset diseases like that, we do think of thalassemia as kind of the traditional ones in Greek and Mediterranean populations. But again, as we've seen in our research studies of healthy people and in our clinic even, you really never know when you sequence even someone who doesn't have symptoms. We find a pretty high frequency of people from all backgrounds who do have something going on that we can act upon.
Yeah, and I've just looked up something on the internet here, and there are some other forms of sickle cell disease that apparently are a little bit unusually higher in Greek populations. So, but also anybody who comes through what's called a founder population. So they grew, their ancestors grew up in a small village where there was any kind of intermarriage. That's something that can be of genetic interest as well.
Of course. You also reference, some of the consumer products that are out there that have been around, 23andMe, Ancestry. So our audience, and I've done 23andMe. I'm sure others have done different things. What how does that compare? Obviously, what you're doing is at a completely different level, but
it's really
how much of what they do is more survey related or origination related versus actual variants, gene variants that are or mutations that potentially would be in your system?
Right. It's a huge difference. I'm not a direct-to-consumer hater. A lot of geneticists, are really put them down. I think they've done a great service democratizing genetics, but there is confusion. Drake, what they do is like taking a book and reading one word every 1,000 words. They are skipping over 999 words, and they're doing a good job of reading that word. What we do is reading every letter and every word of that book. So when they tell you something, it's actually pretty good quality, but they're missing in enormous amounts of disease-associated information. So that's the difference.
Just because you have a mutation or a variant, let's say, right? It doesn't necessarily mean that a disease will develop, correct?
That's right. There's a concept called penetrance, and it's basically a fraction. If I have this change in my DNA, what we used to call a mutation, and now we more of the time call a variant. If I have that change, what's the likelihood? What's the probability that I will develop that disease in my lifetime? And we know that the penetrance of, let's say, Huntington disease mutation is 100%. The penetrance of the BRCA1 for breast or ovarian cancer that unfortunately some women carry is about 65 or 70%. The penetrance of many other DNA changes is more like 40 or 50%. So it's definitely can increase your risk, but is not a guarantee that you're going to get it. That's right.
Right. And for certain ones, I think, there's certain things you can do as an individual, if you do find a variant, but you're not going to be none of them are going to be drastically changing your lifestyle in the moment, correct?
That's right. Often it's actually just telling you to do what you'd be doing otherwise. I mean, we had a guy, and this was me, when I passed 50, I knew I was supposed to get a colonoscopy, but I just didn't do it because I didn't want to bother with it for a year. We had a guy who came into our Preventive Genomics clinic. He was 55 or 6. He found out he had a mutation for colon cancer. Now, the next week, he went out and got his colonoscopy, and guess what? They found a couple of precancerous lesions. So sometimes it's not even, doing anything extraordinary. It's just making sure that the people who are at risk get the kind of surveillance they ought to be getting anyway.
Was the right med test. Mm-hmm. And what that is and what that tells people. In fact, I went through it myself because I thought it was interesting.
Yeah, it's really cutting edge, but it appeals to doctors because intuitively doctors prescribe medicines, and they are used to the idea that a medicine might work better or worse than somebody else. So it's called pharmacogenomics. And it's the idea that the markers, sometimes these hundreds of markers along your genome, may influence how a medication works in you. Does it work? Does it not work? Does it have a side effect? Does it not have a side effect? Do you need to start a higher dose than somebody else with a different marker? And so we've reached the point, this is really transitioning from research into clinical care. We've reached the point where most people think it's starting to be useful. And so we offer this as part of our Preventive Genomics clinic to people, and they can tuck it in their medical record. And then, when their doctor gets ready to start them on a particular drug, you can just check it and say, oh, my pharmacogenomic profile suggests that I should be started on a little bit lower dose of that in order to avoid side effects. Bethany, anything else you'd want to add talking about one of the right med test?
I do think, especially for certain types of medications that are used, like for example, psychiatric medications, that can usually be a really long, what we call therapeutic odyssey, because people react to them really differently and it takes a while to test them out. So for something, especially in that field, it's really helpful to have that as another tool in the doctor's toolbox to kind of take into account when they are prescribing a medication.
Yeah.
So it's really fascinating stuff.
Yeah, it's not an exact thing, Drake. It's not like, you're going to get told exactly what. It's still probabilistic, meaning that, you have a higher probability or a lower probability of side effects. Genetics just doesn't work, in that sort of deterministic way that everybody sort of imagined it might. But probabilities are what we build public health policies around.
Right. Is there anything covered by insurance, and do you foresee that changing in the near future or longer term?
Yes, there's progress on that. For example, genetics has been particularly useful right now in, it's not even cutting edge anymore, in neonatal intensive care units. And just last year, for the first time, even though we've known it was useful for 4 or 5 years, for the first time, United Health Group, which is the largest health insurer, started reimbursing for sequencing in neonatal intensive care. They are, of course, understandably very wary of paying for everybody to get genetic testing. But I am working with that company and several others to try to encourage them to do economic modeling and to actually try this in what essentially are real life experiments. In other words, if you take a region of the country, if you take a certain set of practices and you okay this, what happens to healthcare costs? Maybe you can save costs. If you can prevent enough illness, the logic would be downstream you could save costs. But that's where our American healthcare system, so fragmented, works against us, Drake, because what they tell me back is, well, on average, people are only in our healthcare plan for 2 to 3 years. So if you're telling them you're going to save money after 5 to 10 years, they might say, well, that's not our concern.
Yeah. And is that any different overseas, anywhere else in the world?
It is. Most European countries have a single payer system, so the government's on the hook for the cost. But by the same token, if they could prevent illness 5 or 10 years from now, they would reap the benefits. So the Scandinavian countries are very keen on this. Some of the Asian countries are very keen on this. It's actually a harder sell in the U.S.
True. And then question getting back to COVID. Obviously everything's been accelerated, and it seems like you were able to shift gears and produce some very interesting research in a short amount of time. Looking forward the next 6 months to a year, where would you like to see your work reach a point with regards to looking at COVID?
Yeah, thank you. We pivoted. We took 2.5 of our FTEs and put them on this COVID project, and we have done that for the last few months without any financial support whatsoever. So we do need to find financial support for that. But at the same time, we're continuing with our preventive genomics work. And so I'm very excited on the COVID front that we and others are contributing to our little piece of the pie, which is understanding the genetics of vulnerability. I think this will be, even with a vaccine, people are still going to get the disease, they're still going to get sick, they're still going to get admitted to the hospital, they're still going to die. So I'm very excited about clues to treatment that this will uncover. And then on our mainstream work, our preventive genomics work, we're very excited about both our clinic and about moving this further into the lives of newborn infants. We are trying to develop satellite groups. So if any of the people listening are in different states or they focus on different healthcare centers and you want your healthcare center to be part of the Baby Seek project, we're going to be trying to seek funding from NIH or seek private funding to support that. so that instead of just looking at 500 families that we're funded to do, we can look at 2,000 or 3,000 or 5,000 families and really transform medical care faster than waiting the many years it usually takes to get an innovation in there.
True. Why don't you, if since we're wrapping up, why don't you, if you don't mind, put up that last slide again with all of your contact information. And I'm assuming if somebody has an interest and wants to reach out to you, there's probably also an opportunity to volunteer
Absolutely.
in any of your work as well, it says.
Absolutely. We'd be delighted to talk to you about any genetics related health problem. If you're just interested in our work on COVID, we'd be happy to tell you more about that. We have a couple of manuscripts that are in process, depending on how deeply someone would like to dive into it. We get some of our best ideas from talking to people who are genuinely interested. So it's not an imposition. We would genuinely like to hear from you.
Great. And I think that's it for questions. So let me obviously take the opportunity to thank you and Bethany for presentation today. Obviously, it's very, very informative, and certainly as much as we've been bombarded with, obviously, naturally with everything going on with COVID information on a daily basis, it's great to see the work that's being done in the research end. And the work in preventive genomics that's helping you out as well.
Very good.
And also to your special guest.
That's right. Thank you very much for inviting us. We are really happy to have given you this time, and we look forward to communicating with all of you who are interested in pursuing further.
Great. Thank you again, both of you, and appreciate it. And thanks again for joining us, everyone. Enjoy your evenings.